Hello everyone, in this video we're going to finish talking about cell transport. So this page shows you facilitated diffusion. We already talked about this during class. Facilitated diffusion is very similar to simple diffusion. The only difference is facilitated diffusion has to have a membrane protein facilitating the process. So this could be a protein channel or it could be a protein carrier, but it helps getting the molecules from one side of the cell membrane to the other side. It could be inside to outside, outside to inside. It doesn't really matter. As long as it's high to low concentration, no energy is needed, then that you can call that facilitated diffusion. It says the word specific over here because transport is based on proteins, right? And proteins are specific in terms of what it does, right? So proteins are folded, so the molecules that can travel through the protein and or bind to the protein and get carried to the other side of the cell membrane, this is all very specific. One type of transport protein will allow only one type of molecule to get through it. And then there are two different types. We have the channel protein, as you can see. There's a channel going all the way through this transmembrane protein. And then we have this carrier protein that we'll talk about a little bit more later. So let's take a look at channel protein. It has a hydrophilic passage, right? Because in order for molecules to travel through, these molecules that are traveling through are often hydrophilic or polar molecules. Because otherwise, if it were to be nonpolar, it can just go through the cell membrane directly. So as an example, we have aquaporins allowing water molecules to travel through the cell membrane protein channel, right? So it has the inside of this channel is all hydrophilic, but everywhere else could be hydrophobic. But this part that sticks out of the cell membrane also needs to be hydrophilic. And then we have some different types of channel protein. One example is a gated channel. So gated channels are ones where it doesn't just have a channel that's open all the time. It has a gate that allows the channel to open up or closing. So for example, ion channels means this type of channel protein allows ions to go through. So molecules with positive charges or negative charges can go through the channel itself. So in this case, there are different types of gates. And they will open and close based on different scenarios and based on different conditions. So there are three different types that we're going to talk about. One is called electrical. So that's dependent on the charge inside and outside of the protein. So we'll look at that. Chemical is based on a chemical binding to the protein and the channel protein, allowing to open and close. And then we have physical stimulus. So the first one that we're going to talk about is ligand-gated channel. Ligand is just a molecule, a signaling molecule that can bind to the protein, the channel protein itself. So as you can see over here, this channel protein is closed, but when these neurotransmitters bind to the channel protein, it allows this channel protein to open up the channel. And the result is the ions can now travel through the channel protein. On the right side, you can see this example happening as well. So similarly, we have a G protein. So this G protein is serving as a signaling for a signal transduction, but it's not a channel protein on its own. So what happens is this neurotransmitter will bind to the G protein. This receptor, G protein receptor, will then be activated. And then there's another protein that sends a signal all the way to this ion channel, which allows the ion channel to open up as well. So ligand-gated channel just means there would be some sort of chemical binding to the channel to allow the channel to open or close. The next one is called mechanically gated channels. So these are based on temperature or pressure. So if you think about it, for example, right, your hands there are a lot of sensory neurons on here, right? So you can sense things. You can feel the pressure when you press down onto something. I can feel that the table is pretty cold compared to the temperature of my hand, and I can feel the temperature as well, right? So these feelings that you get of the outside world are, there has to be receptors that are allowing these feelings to happen, right? So there are receptors that are accepting these signals and then transporting that signal to your brain, and you're able to make sense of these signals. So mechanically gated channel, as you can see this definition that I copied off directly from the internet, it says open and close in response to mechanical vibration or pressure, such as sound waves or pressure of touch, right? So what that means is, there are a lot of these receptors in the cells of your hands, for example, or your ears, for example, they're able to transport that signal to your brain because there are these cells that would open up its channels based on these mechanical responses, okay, or these mechanical stimuli, I meant. Okay, so this picture right here is not exactly relevant, it looks like. This might be a chemical pressure, but I hope you understand when I talk about like your skin or your ear, for example, you can feel these touches because these cells are able to cause chemical changes as a response to the feeling, right? So I press on to this table, I can feel it's cold because the temperature is able to open up receptors inside my cells, and then they're able to transport that signal all the way to my brain. The next one is called voltage-gated channel. So this is a typical example will be your neurons. So we'll talk about this in the future. Specifically, we'll talk a little bit more about neurons, but voltage-gated channels open and close based on the difference in charges across the cell membrane. So for example, as you can see right here, right? So there's this difference in charges across the membrane, and that will allow the sodium, this is a voltage-gated sodium channel, it will allow it to open and allow sodium to come into the cell. And then once that voltage changes, then this protein channel, this sodium channel will then close, okay? So you can see with the change in the voltage, the membrane, the membrane channel, the protein channel is able to close or open or close or be activated or inactivated. All right. The next one is called a leakage channel. So a leakage channel is often just open all the time, but sometimes leakage channel can close or randomly open. So these are when you don't have to regulate that molecule that travel through it as much. So in this case, the molecules can just go through the channel pretty frequently or randomly. Okay. So it's still a passive transport, high to low concentration, but it's not as regulated compared to the other ones that we've seen. On to the next, we have carrier proteins. So carrier proteins are not, this one is not a carrier protein, but these two are carrier proteins. So carrier protein goes through the process of you have a molecule binding to the carrier protein, and then this binding will induce a conformational change of the carrier protein, which means the shape of the carrier protein will then change, right? So then the result is now this carrier protein will open up to the other side of the cell membrane, which allows this molecule to move into the cell. And then once this molecule leaves the protein, this protein will go through another transformational change, and then it will go back to its original shape and have its opening facing towards the outside of the cell. Okay? So this word conformational change is something that you need to understand and be able to describe yourself. Conformational change is just talking about the shape of the protein changing. And the shape of the protein changing can be based off of chemicals binding to it, right? So in this case, when the chemical binds or not bind, it will change the protein shape. It could also be dependent on the pH and the temperature in the surrounding environment. On to the next, we have active transport. So active transport is the opposite of passive transport, which means you're going against concentration gradient, low to high concentration. This is when you want to, for example, build up a lot of certain chemicals inside your cell. So you keep putting those chemicals into the cell, even though your cell is already concentrated with those chemicals. For example, glucose, right, is an energy source. We want more and more glucose inside our cells, oftentimes, even when we already have glucose in the cell, right? So you want to pump even more glucose into the cell through active transport, low to high concentration. Active transport is all caused by a conformational change of carrier protein. So you can't do channel protein because channel protein there's not a lot of regulation there, right? You just have this opening that naturally the molecules want to go from high to low concentration and passive transport instead of active transport. So active transport, you need more regulation and open and close the protein, especially carrier protein, based on what is happening in the cell. And because this active transport, you're going from low to high, this requires energy as well, because going from low to high, this is not what molecules would naturally do, right? So you have to put in energy and to maintain this orderness, right? Because passive transport is disorderness, or diffusion is disorder and active transport is order. So you can take a look at this video with me.